The influence of pot-type ceramic candle filters, with and without activated carbon, in the abundance of bacteria in treated water. Abstract: in this research the potential growth of fecal indicator bacteria, Fib, and heterotrophic bacteria in water stored for a 24-hour period, following POU treatment using pot-type ceramic candle filters, traditional or with activated carbon, to treat contaminated and chlorinated water sources. Independently of the source, thermotolerant coliform and enterococcus were rarely detected in filtered water, showing that tested POU devices were effective against Fib. In contrast, heterotrophic bacteria count increased during storage in all scenarios investigated, which indicated that some species from this group were able to grow even when chlorinated water was filtered, although higher counts were observed when contaminated water was treated. Moreover, this increase was about 2 log 10 higher for ceramic candle filters with activated carbon compared to traditional ones. Although Fib were mostly absent in treated water, the outcomes of this research suggests that ceramic candle filters may be suitable to treat contaminated water but not as appropriate to treat well-disinfected water sources, presenting adequate free chlorine levels, as it can enhance the presence of heterotrophic bacteria, and consequently opportunistic pathogens, especially if activated carbon is used as a filling of ceramic candle filters. Keywords: activated carbon, bacterial growth, ceramic candle filter, HWTS, POU water treatment. Introduction. Concerns about drinking water quality has increased the number of consumers that adopt point of use, POU, drinking water treatment as a measure to reduce taste and odor issues, normally caused by undesirable chemicals, as well as to remove waterborne microbes where public water treatment is inadequate or inexistent. 1. POU water treatment can include various methods such as the application of heat or UV, example boiling, solar radiation, UV lamps, chemical disinfection, example chlorine, and physical removal by filtration, example slow sand filtration, ceramic filters. Most of these methods have been reported to improve physicochemical and microbial water quality of drinking water. 2. 3. Ceramic candle filters have gained widespread use around the world as a low-cost and easy-to-operate POU water treatment capable of producing safe drinking water. 4. In Brazil, although the introduction of high-tech water purifiers in the last decades contributed to a decline from 57.2%, 1990, to 51.3%, 2004, in the proportion of private permanent residences that possessed one of these filters. 5. They are still believed to be the preferred method among Brazilians as this proportion was maintained stable from 2004 to 2015, varying in a range from 50.27 to 53.4%. 6. Sponsors. The most common types of ceramic candle filters are pot-type and faucet-type filters, Figure 1. The pot-type ceramic candle filter systems consist of a porous ceramic candle filter attached to an upper reservoir that sits on top of a lower storage reservoir. Source water is poured into the top reservoir, passes through the ceramic candle filter due to gravitational forces and drips slowly into the lower reservoir, in which filtered water is stored. 7. The storage reservoir is normally fitted with a tap and the top one with a lid. In the case of faucet-type filters, a porous ceramic candle filter is fitted in a sealed case, in which water flow through, and that can be installed to a standard faucet. 8. 1. Pot-type and faucet-type ceramic candle filter systems. Imagem 2. Source: Stefani Ceramic website. Ceramic candle filters can be made of various mineral media, including clays and other fine particles. The media are blended, shaped, dried, and fired at different temperatures to achieve required pore sizes and filtration properties. 9. Similarly to other filtration methods, the traditional ceramic candle filters remove particles larger than the size of the pores in the filter material. 10. Moreover, a thin biofilm layer developed on the ceramic candle filter's surface has also been observed to increase particle removal. 11. However, as usage time increases, this layer can become very thick, causing a decrease in the flow rate. At this point the candles should be replaced or adequately cleaned to restore its functionality. 7. 9. To prevent biofilm formation within the candle itself, some ceramic candle filter producers develop new candles with colloidal silver impregnated on their surface or mixed with the media before firing. 9. One of the main advantages of POU ceramic candle filters is that it does not require energy supply and, therefore, they are an attractive alternative to remote rural communities. In contrast, the major risks to the success of all forms of ceramic filtration are hairline cracks and cross-contamination. Even the presence of only slightly cracked filter can make them unsuitable for reduction of particles and microbes from water. 9. Moreover, even when the filtration process is effective, the quality of stored water can be affected by various other factors, including condition and duration of storage, reservoir characteristics and water source. 12. Typically, well-designed ceramic filters present satisfactory removal rates for suspended solids and most bacteria, protozoa and microbial cysts, but is ineffective against viruses since their size is too small to be retained by the filter. Previous studies have shown a turbidity reduction ranging from 80 to 99%. 13, 14, 15. With respect to microbial contaminants, reference 16 observed a reduction of 4.3 and 4.6 log 10 for Cryptosporidium parvum and Giardia lamblia, respectively. Moreover, removal rates varying from 2 to 7 log 10 for total coliform and E. coli have been observed in previous research. 17, 18, 19. On the other hand, ceramic candle filters do not remove chemicals itself. In order to address this, some ceramic candle filters incorporate activated carbon inside the candle, making them capable of absorbing various chemical compounds that presents high risk to human health. According to reference 20, activated carbon can remove some pesticides, herbicides, metallic contaminants, disinfection byproducts, DBPs, organic compounds, as well as free chlorine and other compounds that causes taste and odor in drinking water. Although chemical removal was significantly enhanced with the incorporation of activated carbon, the WHO has long nurtured that POU water treatment based on filters with activated carbon is not appropriate to use in poorly disinfected waters, as it can support the excessive growth of bacteria, and consequently, increase the risk of enhancing pathogen concentration. 21. Moreover, the reference 22 in a recent study using molecular techniques, observed that the use of activated carbon block filters promoted significant alteration not only in the bacterial abundance but also in the bacterial diversity of filtered water. Most drinking water standards require the use of FIB to indicate fecal contamination and heterotrophic bacteria as a surrogate to process control. 23. The Brazilian drinking water regulation requires the absence of E. coli and total coliform in 100 ml of treated water for human consumption. Moreover, in order to guarantee the integrity of the distribution system, it also requires the analysis of heterotrophic bacteria, which may indicate some irregularity in the distribution system if its concentration reach levels above 500 CFU/ml. 24. It is relevant to mention that most of recent researches using POU ceramic candle filters focused on the faucet type. Although some studies considering the pot-type gravitational ceramic candle filters have been found in the literature, most of them were performed long time ago. Furthermore, most of these studies were focused on the efficacy of these systems in removing microbes when applied to treat various water sources, but studies to investigate the effect of these type of filters on bacterial growth were rarely found in the literature, especially when chlorinated water source was considered. When pot-type ceramic candle filters are employed, filtered water is stored in the lower reservoir, normally for a period no longer than 24 hours, as these systems are commonly refilled every single day. As ceramic candle filters with activated carbon is expected to produce water without remaining free chlorine, it is believed that bacteria growth in treated water is very likely to happen during storage. This research aimed to investigate the potential growth of thermotolerant coliform, enterococcus and heterotrophic bacteria in drinking water stored for a period of 24 hours, following POU water treatment using pot-type ceramic candle filters, with and without an activated carbon filling, and considering the use of contaminated and chlorinated water sources. 2. Material and methods. A. Experimental design. An experimental study was developed to assess the potential growth of FIB and heterotrophic bacteria in stored water, following a POU drinking water treatment based on pot-type ceramic candle filters fitted with traditional ceramic candles or ceramic candle filters with activated carbon from a very popular brand, not displayed due to ethical reasons. It is important to mention that all tested ceramic filters did not present any trace of silver, which is commonly impregnated in these types of filters nowadays. According to the candle's producer, the ceramic candle filters for gravitational systems used in this research presented pore size varying from 5 to 15 m. Moreover, contaminated water and well-disinfected chlorinated water sources were used, resulting in 4 tested scenarios. The experiment and all the analyses were performed at the Laboratory of Sanitation, LabSan, from May to July 2020. Figure 2 presents the schematics of the experimental design, including the 2 treatments tested, which varied according to the presence of activated carbon, coconut shell, inside the candle, TAC, or not, TT, and considered 2 water sources, contaminated, A1, and chlorinated, CT30, A2. All POU water treatments were tested using 2 replicates, and therefore, 8 of these systems were mounted side by side on a bench in the laboratory. It is important to bear in mind that the experiments were continuously running during the entire study period, with exception to the weekends, and therefore, fresh contaminated and chlorinated water were daily prepared and added to the top reservoir of ceramic candle filters every weekday. However, as presented later in the section, water quality monitoring was performed weekly. It is also relevant to mention that the ceramic candle filters were not cleaned during the entire experiment period. B. Preparation of contaminated water. As previously mentioned, two affluents were used in this research, Figure 2 contaminated, A1, and chlorinated, A2. Affluent A1 was produced by adding 25 L of tap water, dechlorinated with sodium thiosulfate, 1.8%, and 2.5 ml of sewage into a clean gallon, 40 L. This mixture was well homogenized before use. This proportion of sewage, 1:10,000, was chosen to simulate potentially contaminated water sources, presenting low turbidity and organic matter content but moderate microbial concentration such as harvested rainwater, groundwater, etc. Fresh sewage was collected 3 times a week at a wastewater treatment plant located in Vila Velha, ES. 2 Experimental schematics. Circle. Picture 2. Legend, equals sampling points, A equals affluent, C equals control, T equals treatment, followed by replicate identification. Office art object. To prepare A2, 25 L of tap water was added into a clean gallon, 40 L, but instead of dechlorinating it, bleach, OBA, 2.0 to 2.5%, was dosed when the level of free chlorine in tap water was lower than 1.5 mg/L. This was performed because preliminary tests indicated that to guarantee the maintenance of a minimum level of 1 mg/L of free chlorine in A2, after 30 minutes of contact time, considering the conditions of the study, tap water should present at least 1.5 mg/L of free chlorine. Finally, 2.5 ml of sewage was added to the gallon and mixed well. See sample collection. Although the ceramic candle filters were filled every weekday to better represent the way that householders normally use them, physicochemical and microbiological analyses were monitored weekly for a period of 10 weeks. Samples of contaminated and chlorinated water were collected from the midpoint of the 40 L gallon, at 10 to 15 centimeters below the water surface, following vigorous homogenization. Moreover, following a period of 24 hours, samples of treated water were collected from the tap of the storage reservoir of all systems. In total, 10 samples were collected every week and analyzed for selected physicochemical and microbiological parameters presented later in this section. It is important to mention that all samples were collected in sterile polypropylene containers and sodium thiosulfate, 1.8%, was used to neutralize chlorine when chlorinated water was sampled. Moreover, the tap of all storage reservoirs was carefully disinfected with alcohol 70% and the first 200 ml of treated water from each system was discarded before sampling. All sampling points are shown within circles in Fig 2. D. Experimental control. In this study, 3 controls were selected to investigate the potential bacterial growth in contaminated water, C1, and chlorinated water, following dechlorination, C2, or not, C3, following a period of 24 hours. Sterile containers were filled with contaminated water, C1, or chlorinated water, C2 and C3, and, immediately, stored in the dark for 24 hours. Sodium thiosulfate, 1.8%, was added to C2 container to neutralize free chlorine. Microbiological analyses were performed in all controls and C3 was also monitored for free chlorine. E. Microbiological analyses. All samples and controls were analyzed for microbiological parameters according to the standard method for the examination of water and wastewater 25. The membrane filtration technique using 0.45 m pore size membranes, Unifil, Brazil, was used for enumeration of thermotolerant coliform, enterococcus and heterotrophic bacteria. Samples were filtered on membranes that were placed onto MFC, Neogen, Lansing, USA, M. enterococcus, Neogen, Lansing, USA, or MHPC, Neogen, Lansing, USA, agar for thermotolerant coliform, enterococcus and heterotrophic bacteria, respectively. The MFC plates were incubated at 44.5 degrees Celsius for 24 hours, whereas the M. enterococcus and MHPC plates were incubated at 35 degrees Celsius for 48 hours. The volume of sample for microbiological analysis varied from 1 to 100 ml according to the water quality. Moreover, samples with higher concentration of microorganisms were diluted to a maximum of 100.000 folds whenever it was necessary. All dilutions were performed in sterile cap tubes using phosphate buffer water, PBS, as diluent. 2 blanks to guarantee PBS integrity were carried out at the beginning and at the end of each batch of analyses. F. Physicochemical Analyses. Physicochemical analyses were performed according to the standard method for the examination of water and wastewater 25. All water samples were tested for turbidity, 2130, using a portable turbidimeter, Hatch 2100P, Loveland, USA, pH, 4500 H+, with a pH meter digemed, DM22, Brazil, and temperature, 2550, at the time of collection, excepted A2, which was analyzed only for pH and free chlorine, 4500 Cl/G, using a free chlorine checker, Hanna High 701, Smithfield, USA. Moreover, samples from A2 and C3 as well as from TT and TAC, treating chlorinated water source, A2, were also tested for free chlorine. G. Statistical methods. Descriptive and inferential statistical analysis were used to evaluate the outcomes of this research. As the number of observations, n equals 10, was not large enough, the normality of data could not be assumed. Therefore, all the results are presented in terms of their median, followed by the interquartile range between brackets. Moreover, non-parametric tests were applied to evaluate the data set. Wilcox test and Kruskal-Wallis combined with Dunn's test were used to investigate differences in the median concentration of evaluated parameters among treatments, controls, and affluents. In order to search for correlations in the data set, the Spearman rank correlation was adopted. Box plots and line plots were also used to illustrate the outcomes of this research. All analyses were performed using R version 4.0.026 and RStudio version January 3, 1056-27 as well as some packages, including ReadXL, DPLYR, Dun.test, FSA, Galber, and Plot2. 3 results. A. Affluent characterization. Temperature and turbidity of chlorinated affluent, A2, were assumed to be similar to contaminated affluent, A1. Affluent A2 presented median pH of 7.4, 7.2 to 7.4, and median level of free chlorine of 1.80, 1.53 to 1.84, mg/L, which decreased to 1.54, 1.25 to 1.59, mg/L after 30 minutes of contact time, but never presented concentration below 1.19 mg/L. This corresponds to a median CT of approximately 45 min.mg/L, which was very effective against all bacteria investigated in the study. No bacteria growth was observed in any sample from A2 throughout the entire experiment period. Affluent A1 presented median temperature of 26.0, 25.0 to 26.8 degrees C and median pH of 7.3, 7.2 to 7.4, which was very similar to the values observed in A2. As planned, the affluent presented low median turbidity, 1.02, 0.90 to 1.32, NTU, and relatively high median concentration of thermotolerant coliform, 2.7, 2.6 to 2.7, log 10 CFU/100 ml, and enterococcus, 2.5, 2.5 to 2.6, log 10 CFU/100 ml. Moreover, the median concentration of heterotrophic bacteria was 4.7, 4.6 to 4.7, log 10 CFU/100 ml, approximately 2 folds higher than Fib. B. Experimental controls. No bacteria were observed in 100 ml of sample from C3, which consisted of A2 stored in the dark for 24 hours. The median chlorine concentration in C3 following the 24-hour period was 1.20, 0.99 to 1.31, mg/L. Considering C2, thermotolerant coliform were observed in only one sample at a very low concentration, 0.2 log 10 CFU/100 ml, whereas enterococcus was never detected in 100 ml of sample. On the other hand, heterotrophic bacteria were always observed in C2, presenting a median concentration of 2.4, 2.3 to 2.6, log 10 CFU/100 ml, which was significantly higher compared to A2, p-value equals 1.64 times 10 to the power of -4. Compared to A1, control C1, A1 stored in the dark for 24 hours, presented significantly higher median concentration of thermotolerant coliform, 4.1, 4.0 to 4.2, log 10 CFU/100 ml, p-value equals 1.58 times 10 to the power of -4, and heterotrophic bacteria, 6.7, 6.5 to 6.8, log 10 CFU/100 ml, p-value equals 1.63 times 10 to the power of -4. However, the median count for enterococcus, 2.3, 2.3 to 2.4, log 10 CFU/100 ml, was observed to decrease slightly following the storage period. See evaluation of pot-type ceramic candle filters. The most relevant results of POU water treatment using pot-type ceramic candle filters, considering contaminated, A1, and chlorinated, A2, water sources are graphically presented in Figure 3. Treatment of A2 using these systems fitted with traditional ceramic candle filter, TT, produced water with a median concentration of 0.30, 0.22 to 0.33 mg/L of free chlorine. It is important to remind that A2 was not dechlorinated before being added to the top reservoir, which resulted in the remaining concentration of free chlorine, following the 24-hour period. On the other hand, as expected, samples from treated water using ceramic candle filters filled with activated carbon, TAC, did not present any level of free chlorine during the testing period. The overall median turbidity reduction, considering all scenarios, was approximately 67%. This resulted in water with median turbidity lower than 0.5 NTU for both ceramic candle filters and both effluents, with no sample presenting values higher than 0.7 NTU. 3 box plots presenting the results of POU drinking water treatment using pot-type ceramic candle filters with 2 types of candle filters to treat chlorinated and contaminated water. Picture 3. Legend, A equals A1 for contaminated effluent or A2 for chlorinated effluent, TT equals treatment with traditional ceramic candle filters, TAC equals treatment with ceramic candle filters filled with activated carbon, CFU equals colony forming units, NTU equals nephelometric turbidity units. The microbial quality of produced water presented a higher variation depending on the effluent and type of ceramic candle filters used as well as on the microorganism evaluated. Enterococcus and thermotolerant coliform were not observed in 100 ml of sample from TT and TAC treating A2 throughout the entire experimental period. Treatment TAC, even when applied to treat A1, presented satisfactory removal rates for enterococcus and thermotolerant coliform, which were not found in 100 ml of samples either. However, when TT was used to treat A1, thermotolerant coliform and enterococcus were observed in 1 and 3 out of 10 observations, but in low concentration, 0.4 and 0.1 log 10 CFU/100 ml, respectively. In contrast, independently of the effluent and the treatment applied, treated water always presented higher counts of heterotrophic bacteria compared to the effluent. Higher counts were observed when A1 was used, resulting in a median concentration of 5.3, 5.2 to 5.3, log 10 CFU/100 ml in water treated using TT, which was significantly lower than the median concentration observed in water produced by TAC, 7.0, 6.3 to 7.1, log 10 CFU/100 ml, p-value equals 8.54 times 10 to the power of -5. Similarly, when A2 was tested, the median count of heterotrophic bacteria observed in treated water by TT, 0.6, 0.4 to 0.7, log 10 CFU/100 ml, was significantly lower compared to the median count using TAC, 3.5, 2.5 to 3.7, log 10 CFU/100 ml, p-value equals 1.53 times 10 to the power of -4. In order to compare the growth of heterotrophic bacteria, the absolute value for the difference between the concentration of heterotrophic bacteria in treated water and effluent was calculated. The median increase in the concentration of heterotrophic bacteria observed in water produced by TT was 0.55, 0.50 to 0.60, log 10 and 0.55, 0.40 to 0.70, log 10 when treating A1 and A2, respectively. On the other hand, when ceramic candle filters with activated carbon, TAC, were used, the median increment was 2.03, 1.52 to 2.37, log 10 when treating A1 and 3.45, 2.55 to 3.70, log 10 considering the treatment of A2. The median increase in the concentration of heterotrophic bacteria in water produced by TT was lower compared to TAC in all experiment repetitions, independently of the affluent used. This increase was significantly lower in water produced by TT treating A2 compared to TAC treating A1, p-value equals 3.25 times 10 to the power of -4th, and TAC treating A2, p-value equals 1.71 times 10 to the power of -4th. Furthermore, the median increase in the count of heterotrophic bacteria was also observed to be significantly lower in TT treating A1 compared to TAC treating A1, p-value equals 1.76 times 10 to the power of -4th, and TAC treating A2, p-value equals 1.67 times 10 to the power of -4th. D. The influence of usage time on pH and heterotrophic bacteria count in water treated by pot-type ceramic candle filters. Most of investigated parameters did not present any correlation with usage time, with exception to pH and heterotrophic bacteria counts, which presented strong correlations, considering the 10-week experimental period. The pH level of treated water, independently of the treatment applied, was observed to reduce more than 1 unit in this period, figure 4. A significant strong negative correlation of approximately 0.86, p-value equals 1.29 times 10 to the power of -3rd, was observed between usage time and pH of water produced by TT, whereas a slightly stronger correlation, ρ equals 0.98, p-value equals 1.50 times 10 to the power of -6th, was observed between usage time and pH of treated water using TAC. Considering the difference from the first to the last week of the experimental period, an increase of nearly 2 log 10 in the concentration of heterotrophic bacteria was observed in treated water using TAC. On the other hand, TT was able to produce water with much lower variation and similar concentration of heterotrophic bacteria throughout the entire experiment, figure 5. A significant strong positive correlation, ρ equals 0.92, p-value equals 1.33 times 10 to the power of -4th, was observed between usage time and the count of heterotrophic bacteria in water produced by TAC. For temporal variation of pH in the affluents and treated waters, considering the use of ceramic candle filters with and without activated carbon, during 10 weeks of operation. Image 5. Temporal variation of heterotrophic bacteria counts in the controls, C1 and C2, affluents and treated waters, considering the use of ceramic candle filters with and without activated carbon, during 10 weeks of operation. Image. It is also interesting to notice that the median concentration of heterotrophic bacteria in water produced by traditional ceramic candle filters, TT, was lower compared to controls. This resulted in a significant difference in the median concentration of heterotrophic bacteria observed between water produced using TT to treat A2, 0.6, 0.4 to 0.7, log 10 CFU/100 ml, compared to C2, 2.4, 2.3 to 2.6, log 10 CFU/100 ml, p-value equals 1.76 times 10 to the power of -4th, and between water produced by TT treating A1, 5.3, 5.2 to 5.3, log 10 CFU/100 ml, compared to C1, 6.7, 6.5 to 6.8, log 10 CFU/100 ml, p-value equals 1.63 times 10 to the power of -4th. However, the higher difference in the count of heterotrophic bacteria observed between treated water using TT applied to treat A2 and C2 was very likely caused by the use of dechlorinated A2 to fill up C2 containers and chlorinated A2 to fill the top reservoir of ceramic candle filters. In contrast to that, the use of ceramic candle filters filled with activated carbon, TAC, produced water with higher counts of heterotrophic bacteria compared to controls after 6 and 3 weeks of operation, considering the treatment of A1 and A2, respectively. However, a significant difference in the median concentration of heterotrophic bacteria was only observed between water produced by TAC treating A2, 3.5, 2.5 to 3.7, log 10 CFU/100 ml, and C2, 2.4, 2.3 to 2.6, log 10 CFU/100 ml, p-value equals 0.048. For discussion. Independently of the water source, A1 or A2, treatments TT and TAC produced water with relatively high pH, ranging from approximately 9.0 to 10.0, during the first weeks of the experimental period, corresponding to an increase of 2 to 3 units. However, at the end of the study, the pH of treated water decreased to approximately 8.0. Although such high values have not been detected in previous studies, Similar increase in the pH level have been observed by 28, 29. Reference 29 observed pH increase of about 2 to 4 units when using traditional ceramic candle filters to treat mineral water and water from the public supply in Recife, Brazil. Turbidity was significantly reduced in all tested scenarios, although slightly better results were achieved using traditional ceramic candle filters, TT, especially when applied to treat chlorinated water, A2. The observed median reduction of turbidity, considering all scenarios, was 67%, 59 to 71%, which was lower than the reduction rates of 95% and 98 to 99%, observed by references 13 and 14, respectively. The reason for that may lie on the fact that the affluent used in the study presented much lower turbidity, approximately 1.0 NTU, compared to the water sources used by 13, 89 NTU, and 14, 30 NTU. Furthermore, although pore size of ceramic candle filters used in the mentioned studies were not displayed, it could be also an important factor affecting the removal of suspended particles, consequently the reduction of turbidity. Based on an initial concentration of Fib, varying from 2.3 to 2.9 log 10 CFU/100 ml, and heterotrophic bacteria, varying from 4.4 to 5.4 log 10 CFU/100 ml, in contaminated water, the chlorination process, median CT equals 45 min.mg/L, was very effective against all investigated bacteria, resulting in an affluent, A2, free of thermotolerant coliform, enterococcus and heterotrophic bacteria in 100 ml of sample. Following a 24-hour storage period, enterococcus were not recovered from any sample of chlorinated dechlorinated, C2, and chlorinated, C3, controls, however thermotolerant coliform were once detected in C2. According to reference 30, the chlorination disinfection process is very effective against Fib and most enteric pathogens. On the other hand, a growth of about 2 to 3 log 10 of heterotrophic bacteria were observed in C2. Previous research has shown that some species of heterotrophic bacteria were more resistant to chlorine than Fib31, and were able to form biofilm and grow within water distribution systems 32. In this study, even in the presence of relatively low levels of free chlorine ranging from 0.16 to 0.34 mg/L, TT treating A2, heterotrophic bacteria were able to grow at a rate ranging from 0.2 to 1.2 log 10. However, it is important to notice that the observed growth may have been affected by the presence of a less effective form of free chlorine, hypochlorite ion, formed at high pH levels, condition of the study, compared to hypochlorous acid, which is formed at lower pH levels and is well known to be more effective against microbes 33. The use of chlorinated or contaminated affluents did not seem to affect the microbial quality of filtered water with respect to thermotolerant coliform and enterococcus. Satisfactory removal rates for thermotolerant coliform and enterococcus have also been observed in previous studies 34, 35. In contrast, heterotrophic bacteria were detected in all samples of treated water, independently of the affluent used, contaminated or chlorinated. Higher resistant to chlorine-based products compared to Fib may explain the detection of relatively high counts of heterotrophic bacteria in produced water when A2 was used, although the treatment of A1 produced water presenting much higher concentration. Similarly, the type of ceramic candle filter, traditional or with activated carbon, did not influence the reduction of thermotolerant coliform and enterococcus significantly. However, higher concentration of heterotrophic bacteria was observed in treated water using ceramic candle filters with activated carbon, TAC. The use of activated carbon to treat water has been indeed observed to facilitate and support the growth of bacteria 21, 22. Furthermore, it is also interesting to note that the concentration of heterotrophic bacteria in water produced by ceramic candle filters with activated carbon increased with time of use, reaching a roughly constant level after 5 to 7 weeks, which was maintained during the final weeks of the study. It is also important to remember that treated water was analyzed following a 24-hour storage period, and therefore, these results are not strictly related to the removal rates promoted by the ceramic candle filters, but also related to the sedimentation process that may have occurred during the storage period. Moreover, the reference 36 observed that competition for space and resources could lead to selection of specific bacterial population, and therefore, competition could also have favored the growth of heterotrophic bacteria. Analyzing the results of heterotrophic bacteria in more details, the median increase observed in TT treating A1, 0.55, 0.50 to 0.60, log 10, and A2, 0.55, 0.40 to 0.70, log 10, were significantly lower than the median increase observed in C1, 2.05, 1.93 to 2.10, log 10, p-values equals 8.78 times 10 to the power of negative fifth, and C2, 2.35, 2.30 to 2.60, log 10, p-values equals 8.54 times 10 to the power of negative fifth, respectively. On the other hand, the median increase in the concentration of heterotrophic bacteria when TAC was applied to treat A1, 2.03, 1.52 to 2.37, log 10, was very close to the median increase observed in C1, whereas when TAC was used to treat A2, this increase, 3.45, 2.55 to 3.70, log 10, was significantly higher compared to C2, p-value equals 2.40 times 10 to the power of negative second. It is relevant to mention that the lowest count of heterotrophic bacteria was observed in C3, not detected in 100 ml of sample, showing that an adequate storage of non-filtered but well-disinfected water. Source, presenting relatively high level of free chlorine, 0.80 mg/L, can be more adequate than its storage following any POU water treatment tested in this research, independently of the ceramic candle type. Although the outcomes of this research indicated that the use of ceramic candle filters, traditional or with activated carbon, to treat contaminated and chlorinated water sources were very effective against thermotolerant coliform and enterococcus, the detected increase in the concentration of heterotrophic bacteria, especially when these filters were filled with activated carbon, may be of concern. There is some evidence that heterotrophic bacteria, which include opportunistic pathogens, are potentially harmful to human health. 38. High levels of heterotrophic bacteria, such as the ones observed in the study, is very likely to be associated with higher counts of opportunistic pathogens, and consequently, it may pose excessive microbial risk to users, especially to the ones presenting weakened immune systems such as neonates and elderly individuals. 39. On the other hand, the higher concentration of heterotrophic bacteria could be one of the main factors controlling the growth of Phyb and other enteric pathogens, which in this case, having higher counts of heterotrophic bacteria could be desirable. In a study to evaluate bacterial responses to applied nutrient amendments, reference 40 observed that competition from heterotrophic bacteria suppressed E. coli growth in stream water. In order to support the development of more effective measures to protect public health, more research is needed to better understand the effect of competition for space and resources in the abundance and diversity of bacterial communities normally present in non-protected drinking water sources, example Phyb, pathogens, and environmental microorganisms. The identification of bacteria strains was not the objective of the study, but with the support of colleagues from the Federal University of São Paulo, some selected colonies of bacteria from chlorinated and/or filtered water were isolated and identified using matrix-assisted laser desorption/ionization, time of flight mass spectrometry, MALDI-TOFMS. All species of bacteria were identified with a score higher than 2.0, which was the same threshold to represent an accurate species-level identification used by reference 41. Nearly all identified bacteria were opportunistic pathogens and included Micrococcus luteus, Klebsiella pneumoniae, Bacillus cereus, Bacillus subtilis, Pseudomonas aeruginosa, and Pseudomonas stutzeri. These results agree with the findings of previous researchers, who observed similar species in potable water samples from some rural habitations of northern Rajasthan, India. 42 and from a hospital in Hungary. 43. In order to keep stored water safer, future research should seek for more effective manners to make ceramic candle filters, including the feeding and storage reservoir, less prone to biofilm formation. Moreover, new disinfection solutions that could be applied to treat water stored in reservoirs of pot-type ceramic candle filters at the time of consumption could also contribute to improve effectivity and reliability of these simple low-cost filters, consequently improving public health. Conclusion. The use of POU pot-type ceramic candle filters applied to treat contaminated water were very effective in reducing Phyb counts and maintaining its quality during a 24-hour period, but not as effective in reducing the concentration of heterotrophic bacteria, especially when ceramic candle filters with activated carbon were used. Moreover, although Phyb were rarely detected when pot-type ceramic candle filters were applied to treat well-disinfected water, presenting high levels of free chlorine, 1.5 mg/L, its use does not seem appropriate in such situation. The reduction of free chlorine to very low levels, especially when activated carbon was applied, allowed the growth of heterotrophic bacteria, which may be associated with an increase in the concentration of opportunistic pathogens, resulting in a higher microbial risk posed to users, especially to neonates and elderly individuals. Therefore, the use of traditional ceramic candle filters would be more indicated to situations in which microbial removal is the aim of the POU water treatment, whereas ceramic candle filters with activated carbon should be preferably used if POU water treatment targets the removal of chemical, although it may enhance the microbial risk. On the other hand, any of these filters should be used if water source is well disinfected and presents adequate level of remaining free chlorine to avoid recontamination of drinking water during storage. Acknowledgement. This research was made possible through the support of the program CAPES/CAPES and Degree October 2018, Profix 2018, funded by CAPES and FAPES.